Hello guys. In this lecture, we're going to focus on introducing the topic of biosensors. There's two sets of suggested reading. One of them is from this book, Chemical Sensors and Biosensors, which we have access to via the library. I will also be posting chapter 1 on the track site. In addition, we also have the Biomaterial Science book. It has a chapter section on biosensors, and I recommend that you read it online. I should also mention that some of the slides that I'm using in this lecture were actually prepared by Dr. Shannon Wigan before, and they have been adapted for my lecture. All right, so a sensor is basically a self-contained device that provides real-time analytical information about a test sample. One thing that I want to point out here is real time. So most biosensors ideally, or most sensors would actually provide that information in real time. That differentiates them from other analytical devices that might require extended processing, things like mass spec or an ELISA assay that are going to take a long time. Here with a sensor, we want to ideally be able to provide feedback fast. Another thing that I wanted to point out is when we talk about analytical information, we're talking about giving information about the presence or the absence of an analyte or the concentration of that analyte. And when I say analyte, what I mean is the target species, a microorganism, a cell, a virus, a chemical compound, a nucleic acid, etc. Now, this type of sensor or these sensors can be divided into at least two categories, and we're going to talk about different categories in the next few slides, but the main two categories are going to be physical sensors versus chemical sensors. A physical sensor is one that is used to measure physical quantities such as force, distance, mass, temperature, pressure, etc. And for the case of biomedical sensors, we're mostly interested in flow, temperature, pressure, and maybe electrical potential that would be associated with changes in the physiology of the patient as a result of a disease. In terms of a chemical sensor, it would be a sensor that is used for quantitative or qualitative measure of chemical substances, such as ions, compounds, etc. So we're going to be measuring chemical species as opposed to physical quantities. Now, biosensors are typically considered chemical sensors, although that doesn't have to be the case. They could actually be temperature sensors or pressure sensors even, but typically they're going to employ a biological molecule as the sensing element. That biological molecule could be an antibody, an enzyme, a DNA molecule, or something like that. Now, there's different definitions of a biosensor as we're going to talk about in the next slide. But for the most part, most books are going to be saying that a biosensor is one that utilizes a biological molecule for the sensing element. Okay, as I mentioned, biosensors have been defined in various ways depending on the source. So some of the definitions are that a biosensor is a device that detects the presence of a biomolecule, whether or not that detection is carried out through the use of a biological sensing element, or a device that is used to measure a biologically relevant or biologically derived signal, or a device that senses using a biomimetic strategy, where biomimetic means that we're going to be mimicking or imitating the design of a biological molecule or biomaterial that has been derived by nature that already has been programmed to interface specifically or detect a particular molecule. And so we're going to be trying to imitate that design to use it for detection of an analytical system. Other definitions, or one by the IUPAC, is that a sensor is a self-contained integrated device that is capable of providing a selective, quantitative, or semi-quantitative analytical information using a biological recognition element. And I have bolded this here because this basically agrees with what we had in the previous slide, where a biosensor is going to be defined as one that includes a biological molecule as a biorecognition element or as a sensing element. I would say that personally, I include both those that include these biological recognition elements, as well as any kind of sensor that detects the presence of a biomolecule, even if we're utilizing synthetic polymers, for example, to do so. Now, here on the top, we have a picture from the biomaterial science book, and it's showing kind of the interface and the blurry lines in between the different types of sensors. So a biosensor here on the left would be one that utilizes a biological recognition element, such as an antibody for detection, for example. On the right-hand side is a biomedical sensor, which does not necessarily utilize a biological molecule. It could be something like a thermometer that measures something that is relevant for biomedical applications. And then here in the center is a medical sensors in general. So all of these medical sensors could be of both different types, basically. As I mentioned, in my case, I consider biosensor both a sensor that utilizes a biological recognition element or one that utilizes a completely synthetic strategy, but that also measures a biomolecule that is relevant to medical devices. And of course, for our class, being a material science class, we want to be inclusive of synthetic strategies, of course, that can provide biological information as well. In this slide, we show the different analytes. As I mentioned, an analyte is going to be a molecule that we are interested in detecting, and it's just listing them in order of the smallest and simplest to the largest and most complex. In the case of pH measurements, we're basically interested in the acidity level of the tissues, and the acidity is directly related to the concentration of hydrogen ions in the solution. So basically, for this case, we would require a sensor that can detect ionic species. Electrolytes or ions are basically the next step after the hydrogen ions, and this is basically concerned with the measurement of relevant ions like sodium ion, potassium ion that are associated with physiological processes. From there we go to blood gases such as oxygen and carbon dioxide, drugs and neurotransmitters, which are going to be small molecules. From here we go to hormones that are slightly larger molecules, proteins that are fairly large molecules, usually in the order of tens of thousands to maybe hundreds of thousands of kilodaltons. This includes antibodies and enzymes. From there we go to the next size scale, which is going to be viruses, then bacteria, parasites, and finally whole cells, human cells, including those of tumors, which are going to be in the order of micrometers, maybe tens of micrometers in size. And of course, the complexity increases significantly in that the biological molecule that we're trying to detect is larger in size, has more functionality, and has more potential to be able to be misidentified due to the number of other molecules that might be present that might have similar moieties. Now, how does a biosensor actually function? So a biosensor is going to always have a recognition element, which we also call as a sensing element, and that's basically the molecule that enables interaction with the analyte of interest. That recognition or sensing element requires selective interaction or affinity toward that analyte. Otherwise, you would basically interact with any molecule and provide false positives for your sensor. Again, these are typically known as sensing elements or biorecognition elements or molecules. We're going to talk about what types of molecules those could be in just a little bit. Now, in addition to be able to interfacing with the analyte, we also need a way to be able to transduce that binding event into a measurable signal. So a transduction element is going to convert the recognition or sensing event into a quantitative or maybe even just qualitative signal that can provide us information about the presence or absence of that analyte or about the concentration of that analyte in the physiological sample. Now, the sensing and transducing elements can be completely separate parts, or like completely separate parts that are working independently, or they could be components that are packaged together, or they could be even different parts of a single molecule, depending on the biosensor. All right. Here I wanted to give an example of the nose as a biosensor. So in our case, our noses have an olfactory membrane that is going to be basically the sensing element that is able to interact with the molecules that are present in the air. And so that is our biorecognition element, basically. That olfactory membrane includes bioreceptors that are able to bind to aerosolized molecules in the air. Now, from there, those cells are going to provide a signal to the nerve cells. And so the nerve cells act as a transducer in that now they're going to convert that binding event between the molecules in the air and our cells here to a electrical signal that can then be processed by the brain. So again, the nerve cells act as the transducer in order to send the signal to the brain, and the brain is going to interpret it and provide an output to our body, whether we like it or we don't like that smell. Now, notice that the nose is capable of distinguishing many different chemical substances qualitatively and can provide a general idea of the quantity down to very low detection limits, depending on the molecule, of course. So that provides you both selectivity, which is the ability to identify one molecule from a different interfering molecule, and sensitivity, which is your ability to detect low concentrations of those molecules. And we're going to be talking about those two words in more detail later on. All right, I already basically said that, so let's continue. Here I have another example of kind of your generalized biosensor system, in that we have a biological recognition molecule which has the ability to selectively bind to the target of interest, which in this case is going to be our green diamond. And that biological recognition element or sensing element could be of various types. It could be an enzyme, it could be an antibody. Both of these are proteins that are found in our bodies. An enzyme is like a catalytic molecule that is able to interact specifically with certain molecules and carry out a chemical reaction in the presence of those molecules. An antibody is a molecule that is developed by your immune system that is specific to the target disease molecule, if you would, and that can bind to it, but does not necessarily carry a chemical reaction with that molecule. It just binds it specifically. Nucleic acids have the ability to bind to complementary strands, or in the case of aptamers, which are nucleic acid strands that have been selected specifically to be able to bind to various molecules, then they can bind to whatever the target is. An aptamer is able to bind to either small molecules or proteins or even whole cells, depending on its development. Bacteria and cells can also be utilized as biorecognition elements because they have certain functions that can be affected by the presence or absence of certain analytes that we might be interested in. By the same token, a tissue that is going to contain cells can be a biological recognition element. And at the same, by the same token, an organelle, for example, a mitochondria, might be able to be used as a biorecognition element because some molecules are going to interfere with the respiration process, for example, of a mitochondria, and that can provide us information about what's happening. Or maybe those molecules can interfere with some of the enzymatic reactions that occur within those organelles, and that can give us information about the presence of those molecules or interference. All right, so basically all of these elements, just to reiterate, they impart selectivity to our sensor. Notice that all of these that are listed here are biological in nature. As I mentioned, I personally also consider other biosensors that are synthetic that utilize materials as a biological recognition element. And so here I wanted to go ahead and mention a couple of other things. For example, one of the things is molecularly imprinted polymers, or MIPs. Those are some materials that we mentioned before. And give me a second, I want to go ahead and put some notes in here. So let's go ahead and put a pen. So molecularly, whoops, not successful. Okay, MIP. All right, forget about writing. Molecularly imprinted polymers which we mentioned before, and these were materials that were basically grown in the presence of the target analyte. And when I say grown, it meant that we were selecting specific monomers that we knew would have good interaction with the molecule that we were interested in detecting. And we would take those monomers and put them in the presence of the target molecule. We would then polymerize those monomers together with a cross-linker to form a three-dimensional polymer network that was basically surrounding the molecules that we were interested in. We would then remove those molecules of interest, and we would be left with a three-dimensional network that had voids or holes that were the right size and had the right configuration to be able to interact specifically with the molecule of interest. You might remember that we had some examples in which the authors basically studied the molecule of interest and found certain functional groups that were of interest, and then they would select some monomers that had complementary functional groups or complementary structures that would be thought to interact favorably with those molecules. And then, those were utilized to create that molecularly imprinted polymer. Another type of biological recognition element that is synthetic in nature would be a hydrogel that is, for example, designed to be able to interface with or to be able to swell in the presence or absence of a particular molecule. For example, for a pH sensor, we would be talking about a pH responsive polymer or hydrogel that would swell or shrink upon changes in pH, or a hydrogel that is embedded with a biological molecule, such as one of these, that would also undergo some kind of change to be able to provide a signal. Let's see, what other ones did I want to mention? Oh yes, there are also some synthetic peptides that we can utilize that would be quenched initially, but that upon interaction with a biological molecule of interest might be cleaved in order to provide a signal. And I'm going to give you an example of those later on. All right, so in addition to our biological recognition element, we also have a signal transducer. That signal transducer could be of various types. For example, we could have an electroactive material that basically is an electrode and that can provide an electrical signal upon attachment of one of the target analytes to the recognition element. If we have a pH change, then that pH change would be measured by a pH electrode, and it would provide information about the voltage difference or potential difference relative to a standard. If we have heat that is being detected or that is being generated, then a thermistor would be, or thermocouple would provide the transducing element. If light was the output from this binding event, then a photodetector would be utilized to be able to change that light into an electrical signal. And then finally, if mass change was what had happened, then a piezoelectric medium would be what would be utilized to provide us an electrical signal. A piezoelectric material is one that accumulates electrical charge in response to a mechanical stimuli or mechanical stress. And then finally, once that signal is generated, there's typically going to be a signal processor. And for the case of an electrical signal, what we're talking about here is things like amplifiers, filters, multiplexers, analog to digital converters, linearizers, or compressors. For other non-electrical sensors, we might have different processing, which could be even your brain, for example, if what we're talking about is a biosensor that develops a color and you might be the one who is interpreting that color. But still there would be some kind of signal processing to convert that signal into an actual information about concentration or the presence of an analyte. And finally, we have the output, right? The output, which could be an electrical signal or other type of information as such. All right, so from here, let's see. Let's go ahead and move on. So In terms of the mathematical response of a biosensor, we have basically the sensor response versus the sensor selectivity. Sensor response, what we're talking about is when sample A, which is basically the molecule that we're trying to detect, is going to interact with the sensing element, which is R for receptor. When those two bind, they are going to form a product, which you can think about it just as a conjugate of the two. And notice that I have used here a double-sided arrow indicating that this is a reversible process, because when these two bind, there's not going to be a covalent interaction. All that we have is secondary interactions such as ionic bonding, electrostatic attractions, or hydrogen bonding, or polar interactions, or hydrophobic interactions that are driving them to bind together non-covalently to form a product, which could be thought of as a complex, basically. Because it is an equilibrium process, we have an equilibrium constant, which is basically the concentration of the products divided by the concentration of the starting analyte and the starting receptor. And that Ka provides information about the affinity of that recognition element toward our analyte. The higher the Ka, basically, the higher the ability of those two to come together, and the higher the response of the sensor. All right, from here we go to sensor selectivity. So selectivity is talking about the capacity of the sensor to respond preferentially to the analyte and not to other species in the sample. So here we have an interfering agent B that is still going to be interacting with the same receptor that we utilize to detect our analyte, and they're going to be forming another product, a product that we are not interested in. And so by the same token, we have that Kb now is going to be equal to the concentration of this undesired product divided by the concentration of the interfering agent and the receptor. And now selectivity is going to be defined as the ratio of Ka to Kb, and therefore a sensor that has high selectivity is going to have a high ratio, much greater than 1, indicating that we are much better able to bind to the analyte A than we are to bind to the analyte, to the interfering agent B. All right, as I mentioned, we can classify biosensors in a number of ways. In a previous slide, we already talked about the physical versus chemical sensor definition. We're going to be focusing now on classifying biosensors according to their recognition element, and we're going to begin by talking about catalytic sensors versus affinity biosensors. We can also classify biosensors according to the transduction method, and that would be basically physical versus chemical, which includes thermal, mechanical, electrochemical, and optical. Starting with a catalytic biosensor, what we have is, and I should mention that typically when we talk about catalytic biosensors, we're talking about sensors that are mediated through an enzyme. The enzyme is basically the catalyst that participates in that reaction. So catalytic biosensors are going to be devices that measure transient or steady state concentrations of detectable species that are either formed or lost or used up due to a biocatalytic reaction, so an enzymatically driven reaction. What we have is that S, which is our reactant of interest, and S prime, which is typically a co-reactant, which it's usually required for enzymatic action. These are going to react in the presence of the catalyst, which is going to be our enzyme. And they're going to be producing products, which could be just one product, or it could be two products perhaps. So in order for us to detect this process, this binding event, we can measure different things. We can measure the rate of production of the products. We can measure the rate of disappearance of the reactant or the co-reactant, or we can actually measure the inhibition of the reaction, because the catalysts can usually be inhibited by other molecules, by certain molecules that can bind on the same active spot on the protein. An enzyme is a protein, so if the active site is blocked, then that enzyme is not going to work. And so basically, with this kind of sensor, we might be interested in measuring either the concentration of the analyte S, Or in certain cases, we might actually be interested in detecting the concentration of the interfering agent, which is preventing that enzyme from actually working. And again, to do that, we can measure any of these substances. We can measure the disappearance of the reactants. We can measure the appearance of the products. Examples of biocatalysts include enzymes or whole cells, organelles, or even tissues that include those enzymes. And here I want to mention that if you're going to use enzymes by themselves, those enzymes can be either individual enzymes or a multi-enzyme sequence. Utilizing multiple enzymes that work in sequence can improve selectivity for the target analyte. In addition, those enzymes can be either purified from cells or they can be recombinant in nature. And then when we're talking about utilizing whole cells or organelles or tissues, what we're talking about is utilizing basically the enzymes that are present in this. And for example, that could be in a mitochondria, which if we're trying to detect metabolic indicators or things that are associated with cell respiration, that would be appropriate. Now, we have to think about enzyme activity. Enzymes are proteins in their own right, and so the activity of those enzymes can be affected by a number of factors. The first factor, or the first thing that we need to think about, is the structure of that enzyme. That protein needs to have a specific primary, secondary, tertiary, and quaternary structure to be able to function. The primary structure of a protein is basically the amino acid sequence. The secondary structure of a protein, including an enzyme, is basically local structures that are due to the interaction of parts of the polypeptide chain, typically through hydrogen bonding, and they lead to the formation of alpha helices and beta pleated sheets. The tertiary structure is basically interaction between the R groups, and it results in a three-dimensional folding of the protein or of the peptide chains. And then the quaternary structure, which is talking about the association of protein subunits. So all of those structures come together to form a large protein, which, as I mentioned, is usually in the order of tens of thousands of kilodaltons to hundreds of thousands of kilodaltons. And unfortunately, those enzymes can be easily denatured, where denature means that they are going to be unfolded or folded in the wrong way, in which case they are no longer active. So we need to make sure that we maintain the right conditions, such as pH, temperature, and cofactor presence, for those enzymes to be able to work. All right, one other thing is that we can actually utilize the denaturing of the protein to be able to detect changes in pH or temperature or cofactor or metal ion presence. If you can think about it from that other perspective, that if the enzyme is not working, it might be because one of these things has changed. The most widely used catalytic biosensor is arguably the glucose meter. Just to tell you a little bit about the development of glucose meters, they were actually first developed in the 1950s and 1960s. In the 1950s, several colorimetric sensors that utilized paper when embedded with glucose oxidase and other enzymes were first developed. This would lead to a colorimetric change that could then be observed, and they typically utilize urine as the biological fluid to be tested. Later on in the 1960s, they developed electrochemical biosensors for glucose measurements, and the first system that was commercialized came out in the 1970s, and it was by the company Yellow Springs Instrument. It was an amperometric sensor that measured current, developed through by utilizing this hydrogen peroxide, which we'll discuss in a second. Since then, there's been a lot of development in the field, including the integration of these glucose meters with insulin pumps that can help to have a kind of a closed system of detection and therapy in one single unit. So the most widely used biosensor for glucose utilizes glucose oxidase, or GOX, as the enzyme for biorecognition. That glucose oxidase is embedded onto an electrode, typically by immobilization with the use of either linkers or with the use of polymer systems. And the function of glucose oxidase is to convert glucose into gluconolactone, which is then hydrolyzed into gluconic acid. At the same time that this happens, the reaction utilizes oxygen, and a byproduct of that reaction is the formation of hydrogen peroxide. So there's at least 3 different ways to be able to measure the concentration of glucose from this reaction. One of them is by looking at the pH change that is induced by the formation of gluconic acid. The next one is by looking at oxygen consumption, and the third one is by looking at the formation of hydrogen peroxide. As I mentioned before, we can utilize enzymes either in their purified form in solution, or we can utilize enzymes that are within live cells or tissues to act as biocatalysts for enzymatic base recognition. The benefit of utilizing cells and tissues is that it doesn't require isolation and purification of these enzymes, and so it might be a more cost-effective process. But most importantly, it is that it enables monitoring of metabolic processes that can actually be used for the transduction purpose. Just as an example, here we have a neural cell which is being grown on a multi-electrode array. So you can think about this like electrodes here at the bottom. And if we are trying to measure the presence or the concentration of an analyte that is in the medium that is surrounding this cell, then what we can do is to measure how does that analyte change the neural signal, electrical signal from that cell. And so indirectly, we will be measuring what's happening to the physiology of the cell, which is mediated by the interaction of that analyte with the enzymes inside of that live cell. I recommend that you go and look at this paper that summarizes a couple of the different techniques that are utilized for biosensing utilizing live cells. In addition to enzymatic sensors, we also have affinity biosensors, which are those that involve reversible non-covalent interactions between the analyte and the receptor molecule, or sensing molecule, I should say. Those reversible non-covalent interactions include hydrophobic interactions, ionic interactions, or electrostatic attractions, hydrogen bonding, dipole-dipole interactions, and London dispersion forces. And they can be thought of as a key and lock kind of interaction. The two molecules, meaning the analyte and the binding molecule, have the right shape, configuration, geometry, and chemical functionality to complement one another so that they have high affinity for one another and not to other molecules. Now, all of these interactions, because of the fact that they are non-covalent, they are affected by the conditions surrounding them, meaning the pH, temperature, buffer composition, which basically means the ionic concentration present in the solution. As we already know, for example, we had discussed that hydrogen bonds can be overcome by increasing temperature, right? So that could be something. In addition, a lot of these biorecognition molecules are proteins, and their folding, their tertiary structures can be changed upon changes in pH and temperature and ionic composition. All right. What are these recognition molecules that we utilize for affinity biosensors? Well, they include receptors. Receptors are proteins that are present on the surface of cells. Something like this would be basically on the cell membrane. They are proteins that have the ability to bind to certain targets. Some of these receptors have the ability to mediate receptor-mediated endocytosis, which means that they would actually enable cargo to be transported into the cell through endocytosis processes. Some of these are signaling molecules where when they bind to their target, they will actually have a configuration change inside of the cell to provide a signal to the cell, etc. In addition to receptors, we also have antibodies. So antibodies are proteins that are made by your immune system to recognize disease or foreign matter. These antibodies consist of this Y shape, and on this Y shape, it is these regions which are called FAB regions, or fragment antigen binding. These regions are the ones that give the antibodies the selectivity toward the target. They are known also as the antigen binding sites, and they are specific to the epitopes on the molecule that is being targeted. This other side on the bottom is actually known as the FC region, and that FC region is shared among different families of antibodies. So this does not provide them specificity toward the target, but the FAB region does. These antibodies are typically in the order of 160,000 in molecular weight, so they are large molecules, yet they are able to bind to a range of different analytes. Okay, in terms of nucleic acids can be utilized, as I mentioned, to either be able to bind complementary strands by utilizing hydrogen bonding. As we know, in DNA and RNA, we have hydrogen bonding between adenine and thymines or uracils, and then guanines to cytosines. In addition to that, there's a special class of nucleic acids called aptamers, which are nucleic acid strands of about 20 to 50 base pairs that have the ability to bind to specific molecules. These aptamers are developed through a process known as SELEX, or these aptamers are developed through a process known as SELEX, which is also known as systematic evolution of ligands by exponential enrichment, in which basically they are picked out of a large pool of random oligonucleotides because of their ability to have some affinity toward the target molecule. Proteins, in addition to receptors and antibodies, which are also proteins, here I'm referring to proteins such as avidin, that has the ability to bind to biotin molecules with very high affinity. Another example would be lectins, that have the ability to bind to carbohydrates, for example. And then lastly, I'm listing molecularly imprinted polymers, which out of all of these are the only ones that are synthetically made, and which are made by basically studying the molecule that we're trying to bind to, selecting monomers or polymers that have some complementarity in terms of electrostatic interactions or in terms of molecular structure to the target analyte, and then they are reacted to form a three-dimensional network around the target molecule, as I already had explained. All right, in all of these affinity biosensors, we're going to be forming these complexes that through the reaction of the biorecognition molecule. In this case, I'm listing an antibody, and L here indicates the ligand, which is basically the antigen that we're trying to detect. When these two come together, they form a complex. The K on represents the rate constant for the forward reaction. The K off represents the rate constant for the reverse reaction. Now, these processes are typically categorized or characterized by their affinity constants of association, Ka, or the affinity constant of dissociation, Kd. And I would say that Kd is typically the one that is most widely utilized. That Kd could also be called the equilibrium dissociation constant, and is basically the ratio of the rate constants of the dissociation process to the association process. Kd is inversely proportional to the affinity of that antibody or that biorecognition molecule to the target, meaning that a smaller Kd would actually tell you that that agent has higher affinity for the target molecule or for the analyte. So typically we're going to be looking for antibodies or aptamers or any biorecognition molecule that has a Kd in the range of micromolar to picomolar or maybe even femtomolar. The smaller that Kd is, the better it is. And I would recommend that you guys go to this website to read a little bit more about Kds. Specifically, Abcam is a company that sells antibodies, and so they have a good discussion about how the Kds are measured, what does the Kd value indicate, etc. All right, the second classification of biosensors is based upon their transduction method. Among those transduction methods, we have physical methods and chemical methods. The physical methods are based on a physical property of the sensing element that is affected through the interaction with the analyte, and these are typically label-free signal detection methods. Label-free means that the recognition and the transduction occurs in a single step without the need for added reagents, for example. And examples of physical transductions would be a mass change or the measurement of a mass change utilizing a piezoelectric system, the detection of a molecule by utilizing changes in the refractive index or changes in the dielectric constant of a material for optical detection, for example. And then chemical sensors are those that monitor chemical changes in the sensing element following recognition. They can be either direct or indirect. In a direct measurement, we're talking about the products changing, the concentration of the products changing, or the concentration of the analyte changing as a function of binding. In an indirect measurement, what we have is maybe the formation of a byproduct that could be tracked, or the use of an inhibitor that could be tracked. For example, the peroxide measurement on a glucose meter. Now, in a lot of these chemical sensors, we will need a label for signal detection. Not in all, but in a lot of them, we do need them. And this label is basically, it could be a fluorescent molecule that could enable us to detect the binding event through fluorescence measurements or colorimetric measurements. Or it could be a enzymatic or maybe an antibody that has an enzymatic label to enable us to do optical detection through either luminescence or absorbance methods. Or perhaps if we have an actual electrochemical sensor, then we might need to add an electroactive molecule that would enable us to detect a signal to indicate the binding process.